Yes, yes, you can start.
Yes, sir. Thank you, sir. Sir, your slide is already visible, but that's fine. I'll just start
Yeah, I
introduce.
will be actually writing things also
Okay, sir. Okay, sir.
and presenting. There are questions I can answer also.
Yes, sir. Yes, sir. So we are starting, sir. Okay, sir. So a very good afternoon to all of you. And today we are in the second day of our lecture series, which is on brain behavioral computing at the epitome of AI. So I am honored to have Professor Rotroy with us. He has been a great mentor for all of us for last duration when we have been working. So Sir Arvinder Rotroy is a professor in Department of Electronics, Electrical Engineering and at IIT Kharagpur. He has an extensive academic background, including degrees from NIT Rourkela, IIT Kanpur, and Sambalpur University, as well as postdoctoral research at Purdue University, USA. His research focuses on machine learning, signal processing, and embedded systems, particularly in applications related to human monitoring, diagnostics, and prognostics. He has published 150 referred journal articles and 100 conference proceedings, supervised 50 completed PhD and 20 more are in progress, and he holds 17 patents with additional applications. Professor Rothroy has developed 50 products at various technology readiness levels, TRLs, and successfully transferred 10 technologies. He is also a founder of 5 startups and has collaborated with major organizations such as General Motors, Samsung, Airbus, Boeing, Indian Railways, ONGC, Shell, Cell, and GAL. So these are the few words which we would like to introduce Rothroy sir, though he has been a great guide. And I am grateful to him because he was not keeping well, but still he accepted our request and present for guiding us in the field. So before taking much more time, I request Professor Rotro sir to please go ahead and just talk about the matter on the subject topic. Thank you very much, sir.
Thank you very much, Priyanka. So, as you can see in the screen, so this is all about the inverse problem, EEG inverse problem. So that means you will see in this picture, we are measuring the EEG in the lab, which is a very dense EEG. So, which is about 256 electrode EEG. So we can see that the signals are coming from the surface of the brain, but we need to know what is inside. So that's basically the inverse problem. We are going to talk about inverse problem solution. So if you have any questions, you can interrupt in between. So I am not a clinical guy, so our research starts as signal processing, especially how to get the inverse models correct so that we can get the particle success. So that is the foundation, and it has been enriched by several PhD students who have worked with me at IIT Kharagpur, about 5 to 7, I think, and several master's students. So all their work, in a nutshell, I'm going to talk about because I know the audience is diverse, and I'm not sure how much math I can do, but as you know, we are professors. We do a lot of math. That is our bread and butter. So having said that, so let us go to the inverse problems of each. So let me go to this. So we call it the sparse algorithms for EEG source localization. So that is the, can you see my screen?
Yes, sir. Yes, sir. Screen is visible, sir.
You can see my screen, right?
So this is not in execution mode. Now it is in execution mode, sir. Okay, sir.
So this is all about the problems which I am saying. So this is the basic introduction of linear inverse problems. So basically, AX is equal to B, you have to solve. So if I go into this, you will be more clear. So in this, actually, you are measuring. So if you have some measurements like B are the measurements. So this is the source here. This side is the source. Then it gets mixed up, all the sources, source 1, 2, let's say source n. So you get measurements B1 to Bm. That means your sources which are S1 to Sn and your measurements are B1 to Bm. So that means, and the sources are connected to, these are the source vector which is connected to the measurement vector. So this is B1 to Bm and this is S1 to Sn. So they are connected to by, so we are assuming a linear relationship. First, let's assume a linear relationship. That means there is a matrix A which will, if you multiply by S, you will get B plus some error. So if S is n cross 1 and matrix A is m cross n and B is m cross 1 and this is m cross 1. So this is known, this is assumed, and this is unknown, this is noise. Is it clear to all of you? So this is a typical inverse problem and we call it instantaneous inverse problem. That means the source at a particular time k, it gives rise to the measurement which is k. So A sk, k is the time index which is bk plus ek. So this is called instantaneous mixing. Now given this problem A, knowing if you know this, how to find this. So this is the typical problem. That means you have to solve, that means A m cross n, S n cross 1 and is equal to B m cross 1 plus error m cross 1. So your job is to basically minimize this. B minus AS has to be equal to E. Therefore, you basically try to minimize this B minus AS norm square, which is norm E square. You minimize this with respect to S. So this is the problem. With respect to S. So to minimize this, so this becomes B minus AS transpose into B minus AS. Then minimize this with respect to S. So find S, this is known, find S so that it is minimized. So there are few problems here. One problem is when the number of measurements is greater than the number of sources. That is case 1. That means m is greater than n. This is case 1. In that case, the problem is redundant. That means you can go for least square solution. And the solution is S is equal to A transpose A inverse into P. This is called least square solution. B is known, A is known, the estimated S is this much. This is called least square solution. That means if the number of measurements are more than number of unknowns, so it is obvious that you have more information than the source, you have more measurements, so we can get very nice picture about the source. But unfortunately for brain signals, the number of neurons can run into billions. And your sources maximum could be 32, 64, 128 or 256. So the basic idea of inverse problems in each measurement, the scientists are saying, researchers, that you should have minimum 128 electrons. Minimum. Otherwise, inverse problem does not work. So now we'll go to more about this. So in this case, the case number 2 is, that means in this case, A S is equal to B plus newt, where A is m cross n, S is n cross 1 number of sources, B is m cross 1, newt or error is m cross 1. So this is error or noise, you can say. So this is measurement. So in brain signals or EEG, so this m is far less than n. That means the number of measurements are much less than the number of unknowns. So therefore, you cannot solve it. It will have lot of problem. So what people try to do is they put some constant like sparsity constant, etc. So this Loretta or S Loretta, there is an algorithm called S Loretta that minimizes this S minus B norm 2 plus some mu into norm S. You can minimize this. That means you get something which is minimum norm solution. This is called minimum norm solution. This is so when you put instead of S2, if you write mu into S1, this is called sparse solution. So these are the basis of the EEG inverse problem. We'll get into details. I'll answer questions at any time because you have 1 hour time. I'm just giving you some idea, but to get into the depth of the research, so we need to. So I'm going to into this now. So these are called linear inverse problems. As you can see, y is known, x is unknown. So k11 x1, k12 x2, k11 xn, a1 and xn equal y1. So there are m measurements as you can see. X is number of unknowns. So you can write K capital K matrix, which is a big matrix as you can see here. So this is the matrix. K is a big matrix, which is m cross n. And X is the source and Y is the measurement. Sometimes this matrix is known as lead field matrix. That means how the cortical sources are connecting to the electrode. So that is called lead field matrix, or sometimes it can be derived from head model. That means this tells you the connectivity of the cortex to the electrodes. So once you, these are called head models. From this can be derived, the lead field matrix or LFM can be derived from head models. So this is the inverse problem. Okay. So then if you go to, these are linear inverse problems. We are not going into nonlinear inverse problems now. I will tell it in the end. So this is linear inverse problem. So the problem is well posed if n is greater than n. That means if your number of measurements m is greater than n, that means the more information than sources, so then it is called well posed. And if the solution is not well posed, that means m is less than n. That means the number of measurements is less than the number of sources. So this particular case is overdetermined. This particular case is underdetermined. So if it is overdetermined, you can find out the more equations than unknown, so you can go for least square solution. If there are more equations, because you have more things which are actually measured, so you have more unknowns than number of unknowns. So this is overdetermined system, and if the fewer equations are then this is called underdetermined case. So the case which you are considering in case of brain interface is underdetermined. So inverse problem can be connotated into 3 different problems. One is called a forward problem, where actually you have a true model, head model, let's say, you mix it up and generate the data, then solve the inverse problem, try to match the estimated model with the true model, that is appraisal. So forward problem, inverse problem, appraisal. These are the 3 steps to set up a particular inverse solver. Is that clear? So now, this is what I have already told. You have a forward model, there is an input signal, you got output signal. To estimate the input from the output is inverse problem. Like you are speaking, what is going on in your vocal tract is inverse problem. What is going on in your lungs is inverse problem. You are taking the ECG outside, what is going on in your heart is the inverse problem. Similarly, you are taking EEG on the head, so whatever is going on inside is the inverse problem. So EEG basics I don't want to cover because you already know that there are certain neural activities which are different scales, they generate the EEG signal. So the EEG signal is coming on the skull, which can be measured, and this is in millivolts. So you can put electrodes and very high precision amplifiers to measure. So this happens in 3 levels: microscopic, mesoscopic, and macroscopic scale, where the EEG is generated at a macroscopic scale, and several neurons they are responsible for EEG generation. So which response time is 100 milliseconds. So 1 millisecond is the synaptic current. This synaptic current comes in the form of spike. Then so many synapses, they generate local field potentials, then comes EEG. So EEG, as you can see, is an activity of the brain in a macroscopic scale. That means lot of EEG, lot of neurons, they are generating this EEG. So that means from EEG, if you want to go to individual neurons, it is a formidable job. It's not so easy. So if you look at this single neuron activity is too small, EEG reflects summation of synchronous activity of many neurons and pyramidal neurons, which looks like a pyramid. So they are responsible for the major EEG signal, which is just below the star. So getting to deep sources is not so easy. That means by inverse problem, I can know what is happening to the cortex. That means just the source we have to below the star. So this is called, in other words, solving this inverse problem is also known as EEG source localization. We call it EEG source localization. EEG source localization. It's called source localization. That means you have to find out where is the effect. That means at this point I am having this electrical signal, in which area of the cortex it is getting activated, neurons are activated. So that is why it is called source localization problem. So this is done by, first of all, we have to know the connectivity of these meso or microscopic level to the EEG electrons. That is number one. So that is how you do by head modeling. So you do by anatomical or physiological head modeling, etc. Then you arrive at a particular instantaneous matrix which gives you the head map. So there are lots of issues here. First of all, there are billions of neurons and pyramidal neurons, so it is very difficult to do it at in the neuron level. So there are million neurons which are collectivity or synchronously fire, and this is resulting called a post-synaptic sensory potential, and they go up to magnitude of 10 millivolts with a duration of 10 milliseconds, because neurons are firing at every second, every microsecond, and the voltages are coming in microvolts. But when there is a post-synaptic potential, that means so many neurons they fire together. So when so many, because although brain is a chaotic system, but still there are some synchronization. So because of that, few neurons will coherently behave. So this is all established. Therefore, you get a reasonable voltage which can be measured by EEG. And this EEG is a result of small current dipoles which is occurring because of the neurons in the cortex, which gives rise to electrochemical currents. So these electrochemical currents are known as dipoles. So current loops. So these current loops totally together, they generate what is called EEG. So therefore, we have a source which is distributed by nature. So we call it distributed source model. It is having distributed over the volume, and each volume, just like pixels in image, these volumes are known as voxels. So each voxel, 3D voxels, will have current in three different directions, X, Y, and Z. That means the dipoles can have direction. A current loop means if the current loop is like this, so Ampere's law says that the dipole will be looking like this. Let me just, so this is how dipole looks. So if there is a current like this, current loop, the dipole direction is this. So that means in a voxel, there will be so many small dipoles, just like atoms. So these small dipoles will have resultants. That means there will be small dipoles. Lots of dipoles will be there. So these dipoles consisting of, you can say, a voxel. So in this voxel, there will be resultant dipole in the x direction. There will be resultant dipole in the y direction. There will be resultant dipole in the z direction. So this is I into S, ES. So these are the three dipoles, three directional dipoles of this voxel. So there will be millions of voxels or many voxels, not million, but we can make it 4,000, 5,000 number of voxels. So each voxel will have three-dimensional current loops. Okay. So you can go up to even 10 millimeter to 20 millimeter. 1 minute. I will have to stop somebody because just 1 minute. Just I'll stop somebody. Sorry about that. So I'm taking class from home, so that's why there is some work going on. So I have to stop. So these are the dipoles. So these dipoles result into, so this whole voxel is represented by a piece of free number of current dipoles. Similarly, there are voxel V1 to Vn, maybe Vn may range to some few thousand or tens of thousands or 20,000. So these each of voxel is several V1. So they together they contribute to the EEG signal. So now moving forward, so this is called, so each voxel is assumed to be a probable source of EEG. So you model it like this. So X is the strength of each dipole, as you can see here. So X is the strength of each dipole. K is the mixing matrix, which is always known as lead field matrix, I told. And Y is the electrode measurement. This is the measurement. And this is your noise or error. You can error on model dynamics or noise, whatever. So you need to find out X. Unless you find X, you cannot really know what is going on inside the cortex. So once you know the cortex, with time, what happens to the cortex, if you know, then you can find out the information pathways. These information pathways, if you discover properly, you can find out emotions, fatigue, depression, stress, lot of other things. So this is the whole purpose of doing the inverse model. So the problem is defined like this: Y is the measurement vector of size n cross t. That is t is the time. T is the time instant. Because Y is measured, you can say the Y is measured like this. Y is measured at Y1, Y2, up to Y, let's say T. So these are time instants. That means at each time instant, the vector Y, which is basically the signals of the electron, Y1 to Yn, they are measured. So each instant, time instant. So total Y matrix will be the number of electrodes into the number of time instants. So Y is n cross T. Is that clear? Y is n cross T, as you can see here. And K is the forward matrix, which is sometimes known as lead field matrix, as I said. And N is the number of electrodes on this scalp, that is already known. And M is the number of voxels. That means the number of voxels is X, I mean, number of voxels. So X will be, M is the number of voxels. What is the size of X? It will be 3 times because it is all directions. Direction X, direction Y, direction Z. So K is 3M cross, each time there will be a dipole, so 3M cross T. So this is the whole idea of this equation which has to be solved. So how to solve this? So this cannot be solved because Y, this is number of measurements are not less than the number of, as you can see here, the number of measurements, that means number of electrodes, that is N, and it is much less than the number of voxels, M. Let's say. This is voxels. I think I'm getting a little bit change of notation. This is number of voxels, and M is number of signals or electrodes. So N, the measurement is much less than N. So this particular problem is ill-posed. So to solve this, you have to, in addition to minimizing this error, this is called error square, norm square measurement. You have to minimize something else like sparsity or some other function of X. So this is what you are trying to do. So this is what you are trying to do. You are solving the alpha norm problem here. So this is minimization of this plus this is a function of ऑनलाइन क्लास का आदत छूट गया इसलिए प्रॉब्लम So this is the alpha norm. You can say this is alpha norm. Alpha can be 2, it can be 1, whatever. And this is a function of x. This can be a sparsity norm. So this is a generic setup of inverse problem for each. So you can see this y n cross t. Suppose you take 256 as, let's consider. N is the number of electrons, let's consider the dimensional point. Y is n is equal to, let's say, 256, the number of electrons. T is the time, how many times this time is done? Because you are taking, let's say, 256 hours. You are taking, let's say, 256 hours sampling. So per second, 256 samples, then you are going for 3 minutes. So that means 60 into 3 into 256. So that many, that is the matrix size. So that is equal to what? How much it is? So we can do it here. 50.
Sir, I missed in between. What is this 3? I just missed, I think.
Okay, 3 means there are 3
Dimension.
No, these voxels are having 3 directions, the current loops.
Dimensions.
So
Okay,
each voxel is represented by 3 current.
Yeah,
so it is
yeah.
3 into n.
Yeah.
So
And
3 into
60
n. N is the number of voxels.
And 60 is,
T
sir,
is the number of times
channels.
done.
Times
T is the number of times done. N is the number of electrons.
Yeah, got it,
So
sir.
it is quite, so it is a huge matrix. This matrix is not small. So this matrix is large, and this K is also lead field matrix, which is also large. X is also large. Everything X will be much larger because X is M will be like 20,000. So 3M will be 60,000. So like dimension of X, the number of voxels is 20,000 into 3 into number of time stamp. That means 60,000 into number of time stamp is this. 250 is 120, maybe around 250 into 120 thousand. That is equal to 30,000. About 30,000. So this is the size of the matrix X, and this is the size of the matrix Y. So this is a large and also a sparse problem. This is called a large and sparse problem. So this is where we come bring in our research to make it quick and also make it accurate. So that is the starting point of the research. So here the number of sources M outnumbers the number of measurements N, thus taking problem ill course. That's what it said. I have already told. So therefore, this problem is close. This M is very large. So therefore, it is not so easy to do. So therefore, what you can do is, so let me see one problem, inverse problem. So this is called the Loretta. This is called standardized low resolution impedance tomography, S-Loretta. This is a very standard algorithm. So how it is done? So this is the alpha norm. This one is norm. Some norm. This is some norm. It can be 2. It can be 1. Or this is 2 1 or 2 2. So whatever. This can be 2. This can be 2. A can be 2 2. So if you solve this one, you get the solution to be like this when A is equal to 2. So the K transpose X. So this is what is T, and then you can find out what is X. So this is not T, this is X. This is X structure. This is X. This is X. This is T here. So with a nutshell, this is the, you can see, these are different lobes, you can see. So in these lobes, these are these points which you see are voxels. So these voxels are computed from the electrodes, outer electrodes. So outer electrodes, you can see it is here. This is the outer electrode. You can see here. These are the outer electrodes. Let me enlarge this. These are 256 outer electrodes. From these electrodes, you are essentially getting these values, and this is at one point of time. So what will happen? The axial, frontal, and sagittal lobes, as time changes, it will be energized differently. So here there is a catch. The catch is, with time, everything is not energized. Only few places are energized. So therefore, it is sparse in time. That means every time you will not get everything activated. So this is sparse in time. And at the same time, all lobes are not active. So therefore, this sparse in space. So it is sparse in time, sparse in space. So therefore, you should have strong sparsity norms to do it. So I shall go to some results. So this is all about the inverse problem. I have tried to tell you what is an inverse problem and how difficult it is, but I have not got into the depth of the inverse problem because these are linear algebra stuffs like that, and we write our own algorithms. So my student Teja Manipuri, who is I don't know whether he's here. He should have joined. He has not joined.
I'm
Anyway.
here, sir. I'm present.
Oh, you are here. Okay, okay. So he has invented a new algorithm. So the name of the algorithm is, you can tell, Teja, what is the name of the algorithm?
Uh,
It's
the
a
name of
complete
the
physical
algorithm
algorithm.
is certainty-based reduced sparse solution that is specifically for high-density EEG electrodes.
Yes. So that is certainty-based path solution, which is completely new algorithm, which is definitely going to change the game because people will do it. But inverse problems are not popular among Indian scientists. It is not. It is only restricted to signal processing community. Like people who are doing clinical EEG, like you from Dr. Priyanka and others. So they don't do inverse problem because first of all the electrodes and the measurement device is very expensive. So unless we do the inverse problem, we cannot do what is happening in the cortex. So if you don't do inverse problem, then what are you going to do? So let me just let me just finish this presentation and I will move to some other thing. So if you don't have inverse problem, then what are you going to do? So for example, suppose you want to study sleeping patterns. You want to study how somebody is sleeping, or you want to study meditation. So basically, if you don't have much electrodes, you have only few electrodes like 32, 64 like that. So what essentially you try to do is you take the electrodes. So this is the number. So these are electrodes. So try to find out how these electrodes are correlated, because eventually you have to find out what is known as information pathway. So information pathways, before that you have to know causality thing. For example, we are doing eye blinking. So that means it will start from your occipital lobe. So that means if you are thinking where from it is starting. It is starting from emotional brain or logical brain or cognitive brain. So you have to know where from it is starting, and you have to know find out where the information is going. That is not so easy without the inverse problem. But if you don't have, you have nothing to do but find out the coherence between different electrons. So that means people try to find out coherence. Coherence is a linear spectral thing. People better find something called synchronization. So that means if you find out the synchronization between different electrodes with time, then you can find out the connectivity. So it leads to something called connectivity. So they do connectivity analysis. So connectivity between different brain areas. Though it is approximate, it is not a problem. So this connectivity changes with time. So when it is more connected, more synchronized, people say it is you are going to sleep or your brain is more focused. If it is chaotic, that means your brain is diverging, the connectivity will be lost. So therefore, there are certain entropy measures of the multivariate signal. This is a multivariate signal. It's not one signal. There are, let's say 19 electrodes, there are 19 signals. There are 19 signals. If there are 64 electrodes, there are 64 signals. So it's a multivariate signal, xk, which is xn, you can say n is the time index. So this, as it varies, you can find out the correlation between synchronization between different, because EEG itself is a chaotic signal, it is non-linear, because the neuron itself is non-linear. So since it is an aggregate of many neurons, so what happens? This EEG signal is non-linear by nature. So therefore, you cannot do it by coherence and correlation. So you have to go for synchronization. There are different methods of synchronization. For example, sin t and sin 2t, they are not correlated, but this is synchronized. Sin t and sin 2t, if sin t starts, sin 2t has to start. They are synchronized. So people go for synchronization measures and they find eventually connectivity, and the connectivity as it changes with time, they decide the behavioral pattern of the brain. So they define certain terms like small world network, connectivity pattern, mean path. So all these things are. So one of the PhD students did a connectivity. So based on this, we have built certain products, like we have built some 2-channel EOG devices. So I shall show you right away. There's a product which is used for alertness monitoring. You can see this is a device which is based on three electrodes basically. You can see this, right? You can see my screen.
Yes, sir. Yes, sir. So what electric positions are temporal, sir? Or means what positions are used here in this device, sir?
So there is one electrode here, one electrode here, one electrode here. So there is essentially three electrodes. So this measures the differential signals and we measure what is known as EOG. So EOG is a part of EEG. So this tells you so this difference between these two, this EOG and there is a battery here, there is a processor here, which Bluetooth communication. So this connects to the cell phone. So you can put a psychomotor station on cell phone and you can find out how your eye is behaving, from which you can find out easily what is cognitive load on you. So cognitive load, alertness, etc. can be found out on the eye parameters like saccade, fixation. Okay. So this is the device which we built it. So this is again based on EOG. So I am almost there in the end of the lecture. I think I would invite some more discussions if it is possible, if there are people available. It seems everybody is looking at the streaming. So there are still 13 people I can see. So if they have any questions, you please ask so that I can discuss and show things which are relevant. I can show some more results also based on our experimental meditation. So, but before that, you guys please ask some questions.
So do we have any question from audience, please?
Hello, am I audible?
Yes, yes, you
Please
are
go ahead.
Yes, so sir, I had one question. That just
Can you please introduce yourself?
yeah, I'm Akanksha Subudhi. I am working in CDAC under Dr. Priyanka ma'am. So
Please go
sir,
ahead.
I had one question that regarding the inverse sparse connectivity. So sir, there might be coming some problem of volume conductivity as well because of taking many electrodes in
Yes,
one
that's a very
particular
good question.
region.
I'll request Teja to respond to. Teja, are you there?
Yes, sir.
Yes, yes,
Yes.
please respond
So,
to this.
in case of EEG, when we are using EEG, so the current, so it has to pass. There are some issues, right? So each tissue, each brain tissue, like the cortex and the scalp has different conductivity levels. So which actually like skull and all. So skull has due to the conductivity of the skull, like the EEG signal is attenuated. So so the point is like you have to like volume conduction has to be considered for EEG, but when we consider the MEG case, so it is just direct measurement of the magnetic field produced by the dipoles. So it doesn't include any kind of volume conduction. So when solving the problem, when we are designing the head model, you have to incorporate the volume conductivity values. So generally three concentric spheres is considered. One is cortex, the other is skull, and the other is scalp. So you can add two more. One is the cerebrospinal fluid, and also the other is the dura mater. Okay. So these 5 levels you can add.
Okay, so just one doubt, like sometimes some like the dipoles, right? So there might be some dipoles positioned in between two electrodes. So that is also a part. So how do you take care of that constant?
Yes. So there is something called Okado constant. So which tells that what should be the number of voxels in the brain that maximum we can go and minimum we can go. So
Okay,
basically speaking, like there are shallow sources and deep sources. So you are right that shallow sources will be more evident on the scalp and EEG. So compared to the deep sources, because deep sources are attenuated because of this volume conductivity.
okay, yeah.
Yeah.
So
So
you're talking about the ones that are closer to the surface of the skull.
yes,
That
yes,
would be
yes, yes.
okay.
So we will make sure that all the dipoles are properly modeled and they are inside the brain. So if we go to the complexities, again, we have to consider the gyri and sulci. So, and then the dipoles are positions, but for the time being, that's how we can conclude.
Okay, thank you.
This inverse problems will have many uncertainties. The first uncertainty is the voxels. When you put things in the voxels, there are many averaging effects already you are doing. And about the conductivity, you construct the lead field matrix as Teja said, using the three concentric spheres. And the conductivity is not constant, it will keep on changing. So even then, the inverse problem, whatever uncertainty is carried because of this lack of information and approximation averaging, is better than the Skal B. That's the point. And it has a better time resolution. That's why it is better than MRI. If you have a dense electrode system like 256, and if you take fMRI, you can only do few things. But in this, actually, you have a neural pathway which can study more finer things like, you can see emotion, arousal, etc., which are more finer. But still, there are a lot of uncertainty, as you are rightly saying, because your head model is not accurate. But it is still better.
Yes, sir. Thank you, sir.
Better than the Skal B.
Yes, sir.
So any other questions, please?
We are inviting questions from other audience as well. Any question, please? Sir, my question is about that inverse problem is that if we are not talking about high volume, high density network connectivity channels, if we are working with a few channels, so I suppose that problem is it making any significance impact on the, if we are working with 2 channel or 4 channel devices, we are working only with few channels in our study?
So the first Professor Rautre has done is the whole head model. Okay. The second one is just localized to certain areas of the brain. So few electrodes and few sources, which sir has done in the previous slides.
Sorry, I'll answer. My mic is mute. But yeah, few electrodes inverse model is out of question. Which I showed you, it was not, it is just a device which is made out of the same essence of EEG. That means you can make your own electrode, that's what I meant to say. That means this device we made ourselves with gold-plated electrodes, it is working. But that is nothing to do with inverse model. We cannot do inverse model below a certain number of electrodes. Otherwise, it will be too localized. It will
Yeah,
not
that
be
was
accurate.
my question, sir. That was my question. It means for low number of channels, we would not be needing to solve this inverse problem. This was my
It
question,
is
sir.
not possible to have inverse problem.
Yes, 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 yes.
You have to do only the average thing. What is happening to the brain on the average? At best, you can make a connectivity study between different areas. That's all. So as you
Got
can
it.
see the screen, we conducted experiment on relaxation, about 200 subjects initially and lot more. You can see it's an international place where a lot of people from all over the world came. So we put them the EEG. This is not a 256 cap. This is 32 channel, 19 channel precisely. So we did some experiments. So with this call, actually we did multimodal. That means a lot of other signals also we took. For example, we took thermal imaging. We took voice patterns. We took the ELISA kit, that is saliva analysis, neurotransmitters. Then eye saccades. We also saw the EEG. We saw all these things together to know what is the effect of relaxation on somebody. So as you can see here, I will just go in the sequence. So you can see thermal imaging. Here you can see thermal imaging, the neurotransmitters, blood flow pattern, like facial blood flow pattern from thermal imaging. Then you can see the eye saccades, voice patterns, everything together we have taken. So if you see the EEG for non-meditators, this is called how the energy is looking at. This is called coherence graph at different frequencies. So this coherence graph of different frequencies with respect to different electrodes, you can see F1, F2, these are electrodes. So the coherence between different electrodes are placed here. This is for non-meditators. Then you can find the same coherence for meditators. We found a remarkable difference that there are correlations coming out. because of deep thought processes. Meditation is not actually thought, but people generally, the common people will think. So these deep thought processes, when you meditate, close your eyes, the deep thought processes connect different electrodes. So coherence is very much visible. So there is no such coherence here when somebody is looking at open eye, etc. Many thoughts are coming, but here there are synchronized thoughts. That's why the coherence is coming. But the meditation ultimate aim is nothing should come up, but that we have not achieved. So these are some of the results as you can see. So this is the experiment which we did at a particular ashram. Then we did the EEG studies on about 200 subjects. So this is all what we have done. I think I have come to the end of my lecture. Thank you, Teja. Thank you very much. If you have any questions, we can answer. We have lots of publications. Teja has a very beautiful publication on this part. We have at least 5 to 7 PhDs they have worked on. The first PhD worked on alertness, that is what happens to the driver synchronization, likelihood. Then second PhD worked on the information pathways. Then third one used on visibility graphs. Then the fourth one worked on coherence, as you can see. The fifth one, Teja worked on the sparsity inverse problem. So I remember these 5. I don't remember if there is anybody else. These theses are with me. So you guys, if you are. We have done considerable work on signal processing, but not on the clinical side. Thank you very much.
Yeah, thank you, sir. This is very deep thought process and very insightful session. I invite questions from the speakers to please come up with your questions so that sir would be able to answer and solve our queries.
Not only myself, I have the expert Teja here.
Dr. Teja as well. I thank you very much, Dr. Teja, as well, for being present in this session and provide the answers of.
I learned from Professor Rautre only.
Yeah.
I learned many things from Professor Rautre.
So I'll introduce Teja. Teja is a BTech from IIT Kharagpur. He's a dual degree. Then he did his masters and PhD here. So then he went to US for about a year. Now he's back in India because he did not like US. He wanted to work here. So he's back now. He'll be here. I think Dr. Priyanka, I can connect to him.
Yeah. Dr. Teja, I definitely would like to have connect with you because we need to learn lots of guidance from all of you, such as senior guys and working so deeply in this domain.
Sure.
And thank you for your presence. Thank you for your presence over here.
And Sridhar Delhi is located at the center, so you will be visiting. Try to visit when you come to Delhi. You try to visit Dr. Priyanka.
Will exchange the contact number as well, sir. I would request you to share the contact of Dr. Teja so that I will connect him.
Teja is now settling. Once he settles, I will connect him to you.
Sir.
Sure.
Any questions from the other team members? Please, you can ask any questions. If I don't know, then I will come back with the answer. I'll ask Teja or we'll come back with the answer. If you know, then because we are all research, many things we really don't know. We are the signal processing people. We know little math, but we don't know what happens in the EEG, how it is generated. So all those stuffs we are not aware of that. So you can ask any questions as far as algorithm processing is concerned.
Hello, sir.
Yes.
I want to know about the ill-posed nature of EEG.
Introduce yourself, Soumya. You introduce.
Yes, sir. Soumya is this side. So I want to know the ill-posed nature of the EEG. What is it? What is this?
Ill-posed. Okay. Suppose
Yes,
you have
sir.
X is the number of unknowns. A is a matrix. What is your background? Are you an engineer or some? You know little math?
Sir, working on BCI and I'm engineer in.
You have basically signal processing person. Right. Okay. So you know little math, right? So, you are actually a signal. You know little bit of math. So I'll tell you what is ill-posedness.
Yes, sir. Yes, sir.
Look at this equation. Ax is equal to b. Okay, plus some error noise, whatever noise is small. So let's say b is known, a is known, x is unknown. Suppose mu is equal to 0. If ax is equal to b, the solution exists when a is n cross n and full rank. That is the number of unknowns is equal to number of equations. So this is case 1. That is the number of unknowns is equal to number of equations. So the exact solution exists. Suppose ax is equal to b plus some noise, where x is n cross 1, a is m cross n, and B is m cross 1. That means the number of equations is m and number of unknowns is n. So this is number of unknowns. And this is number of equations. A is known, B is known. In that case, number of equations are more, so therefore you can do a least square fit. That means you can just do Ax minus B norm square minimize. If you minimize, you will see that X will turn out to be, this is called least square solution. Very nicely done. This is the solution. This is the solution. For case 2, when the number of equations is more than number of unknowns. Now this is case 2. And case 3, that means Ax is equal to B plus epsilon. The number of, that means A is m cross n, X is m cross 1, and B is m cross 1. So if m is less than n, that means the number of equations is less than number of unknowns. If the number of equations are less than number of unknowns, there will be many solutions, not just one solution. That is called an ill-posed problem. So this problem is called ill-posed. That means the number of electrodes is lot less than the number of sources you want. So this is called ill-posed, ill-posedness. Are you getting my point?
Yes, sir. Yes, sir.
So in that case, you have to not only minimize this X minus B norm, this minimization will have many solutions. Many solutions will minimize this, so you have to add additional constants like minimum norm solution. That means you are not only solving the error square but also going for the minimum. So if you change these 2 to 1, it is called sparse solution. So those are actually a little bit higher stuff. I can tell you in a leader anytime you want. So ill-posedness is when you have number of electrodes you are measuring is much less than the number of unknowns which is inside. Alright, so this is called ill-posedness.
Yes, sir. Okay. Thank you, sir.
Welcome.
Any other question from the audience? So there is a question, is there, the person can come online as well. Can ask who was put this question.
Hello.
Yes. Please go ahead. Please go ahead.
Hello, Kasdeep this side, sir, from CDEC Delhi team.
Yes.
Sir, I want to ask about the how to determine the quality of the signal from the EEG device.
Quality of signal.
Yes, sir.
I think this is a very good question, Akash, and I think this is
quality of signal. See, quality of signal is whether the EEG is touching the scalp or not. So there
Nice,
are
sir.
in that quality.
That is touching the scalp. Usually we use the eye blink or jaw clench for our quality check. What
Yes.
else, or is there any predefined algorithm for it?
So you mean to say how to check the quality of signal for the inverse problem, no? That's what you want to say. Inverse after solution.
Sir, I'll just interrupt in between, sir. I think, sir, Akash wants to understand if we are having 2, 3, 4 more devices, and if we want to compare the quality of EEG, so how we can by the technically how we can qualify the EEG quality? How can make a baseline understanding the quality of EEG?
That means you have got 5 devices. You want to know the EEG quality. So that is done by spectrum analysis.
Yes, sir. You the same question you want to ask, Akash, or something else?
Yeah, yes.
No, he's for us asking for the quality of inverse solution. That's what you want.
No, sir. Quality of EEG data,
Yeah.
EEG signal.
So if there are 2 signals, 2 voices, let's say, how to find out the quality which is better? So there is a predetermined notion that this is the frequency. Suppose you close your eyes and measure EEG by 1. Just close your eyes. So then other person will close your eyes. Other person, 5 people, you are getting 5 different EEG devices. So once the EEG comes, the first check is do the spread spectrum. That means you take the autocorrelation matrix, find out the spectral spread. If it is too much spread in the spectral, it is too noisy. That is number 1. The second, if there is a spike, if there is a lot of spike, the spectrum will be flat. If the spectrum is flat, then the EEG quality is not good. So that is one way. So then there is estimation of SNR, signal to noise ratio. So because as you know, EEG maximum 30 Hz or 25 Hz or less than that. So this part spectral density band should be dominant. If it is not dominant, then there is some problem. So these are some of these checks. If you are doing the experiment on the same condition, these are the checks you can do. And as you said rightly, if you blink the eye, the artifact will be very clear. If that is not happening, that is also quality check.
So in addition to what sir told, in general, how it is done is like you take a sphere, you know the conductivity of the sphere, you fill it with some fluid, and you insert manually some dipoles inside that sphere and try to put EEG electrodes around it. This is a quality test. Like, so we know how much is the amplitude of the dipole and everything we know. So how well this an inverse problem, an inverse solver is chosen, and then it is solved and trying to match. So that is also another way of understanding
This
the
is a much
quality.
better way actually. I did know this. Ravi,
Yeah,
you are
it's very
actually
impressive
creating
as well. Yeah.
But yeah, sometimes cost issues also, I think. We have to fill in with the conductive fluid, which is very near to the cortex. And there is a glass kind of bowl, which is around the conductivity of the skull. And there is manually a dipole that is being inserted.
Yeah, thank you. That's a very nice answer.
Okay.
Yeah, very nice answer.
More questions, please.
Do we have any other question from audience?
One common question is how do I do inverse modeling? Is there a software or how do I do it? Generally, if you get very high channel EEG, they will give you their software for inverse modeling. Accelerator is a common thing. Otherwise, Teja has a software, he'll give it to you.
Yes,
I
yes.
think you can build your own software, it's not a big deal.
Yes. So great. I think this been a very insightful session, sir, and we learned very take home messages over here. There's lot many things to at least to us to know that lot many mile ahead to go before this leaves. So lot many things we are not
Yeah,
aware
actually the first thing CDAC should acquire is a 256 channel EEG machine.
of. Yes,
Because
sir.
you are doing so much neuroscience there, a lot of interest there. So if you guys, because I got one 256 channel, it was damaged. Now it is out of service. You cannot fix it. But in your case, because it is continuously being used, I think you guys should acquire one 256 channel. It will be around 1 crore right now, 80 lakhs or so. So
Sir.
that will give you a lot of insight to a lot of things which has not been explored now.
Got it, sir. Got it, sir. Yeah. So thank you very much, sir, for your time and guidance. At the same time, thank you to Dr. Teja as well to come on board and giving us so many inputs on the solution. Definitely, we are going to connect you as well. So, sir, you took out the time when you were not well as well, and still you had interaction with us and guided us on the subject matter. So I'm really grateful to you. And apart from this session as well, when we get your guidance all the time during the projects and off projects as well. So you are a true mentor for all of us. So thank you for your teaching enthusiasm, sir. You always teach us with so much of enthusiasm. So thank you, sir, for your time and effort, sir.
Thank you. Thank you.
Good.
God bless. Thank
Thank
you, Teja. God bless you, Teja. Bye.
Thank you.
Thank you, sir. Bye.
Thank you. Thank you. Thank you.
Thank you, sir.
Thank you all. Thank you all.
Thank you.
Thank you.